Hi, this is going to be the Edexcel Biology Unit 1, May 2024 paper. So this is a small series that I'm doing. It's where I'm solving past papers with complete answers, with complete model answers, and with some additional notes for revision, just so that it can help students whenever they're revising to properly revise the whole syllabus. Basically, like, what I'm trying to aim with this series is that I want you to understand how to solve the question. I want you to understand the concept behind the question. I want to remind you about what or from where we're answering the question from, like which chapter or which part of the chapter we're talking about. So along with the questions, I've also written down some notes and I will explain some things as I go along. So yeah, let's start, okay? Question 1, enzymes are biological catalysts. Enzyme molecules are formed from chains of amino acids joined together. Which pair of elements is found in all amino acids? So over here, as you can see, I have drawn a small diagram of the molecule, right? So over here we have the amino group, right? Over here, this right here is the variable R group, and over here we have the carboxylic acid group. So these are the components to it. And an easy way that I remembered the components of protein is I memorized this over here. CHON. 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 Carbon, hydrogen, oxygen, and nitrogen. Again, we have nitrogen here, hydrogen over here, carbon right here, oxygen here. I think that's pretty easy to understand. So yeah, I kind of made, like, whenever I was studying for my exams, I made notes, and protein was always CHON, carbohydrates I think was CHO. Yeah, it's easy to understand. Like, one easy way to understand or to remember is that proteins has nitrogen. Compared to carbs and like fats, protein is the only one that has nitrogen in it. So yeah, the answer here is going to be A, carbon and oxygen. Moving on to B. So which row of the table describes how two amino acids are joined together in an enzyme molecule? We're talking about amino acids over here, which, if you remember, amino acids are components of proteins. So it's not going to be an ester bond, so the first options are wrong. However, it is going to be a peptide bond. Let's see though. The answer is going to be C. It's going to be a peptide bond. Reaction that forms it is condensation. So again, amino acids are peptide bonds. Condensation reactions, they build, which means that they join two molecules together, right? I'm so sorry. Yeah, they join two molecules together. Hydrolysis reactions, they break bonds. Therefore, the question asked us join together. We're going to choose C, since it's condensation. Moving on to C. Describe how a chain of amino acids can form an enzyme molecule. Right? So, a chain of amino acids, which is the primary structure of a protein, becomes a secondary structure. The amino acid chains, they fold into either a beta helix, chain, or a beta pleated hydrogen bonding. Secondary structures become tertiary. Secondary structure. The secondary structure becomes a tertiary globular structure with an active site. Again, enzymes, they are globular proteins and they are compact. The bonding within them, just to revise and remind you, we have hydrogen bonding, ionic and disulfide bonds between the R groups. Here's a small little diagram that I drew of the enzyme. Honestly, I kind of wrote this answer short, but you can add in whatever you want into it. I just wanted to make it understandable for you. Right? Now let's move on to question 2. Question 2 is about DNA replication. So let's see. Errors in DNA replication can give rise to mutations. The diagram shows part of a DNA molecule. So over here, we've got a DNA molecule, and we've got some labeled pieces. The base labeled Q is thymine. So which is the base labeled P? Over here, we have Q right over here. So this is thymine. Which one is this one going to be? Over here. This is an easy way to remember it. Thymine goes with adenine, and guanine goes with cytosine. How do I remember this? Or when I was studying, how did I remember this? I just remembered GC, group chat. So if you remember one of the combinations, you can pretty much just guess the second one. Since G is with C, you're left with A and T. So, you just put 2 and 2 together and you get the answer. It was easy. So yeah, the answer is adenine. And over here we have uracil. However, uracil is only present in RNA, not in DNA. And uracil is what replaces thymine. Okay. So which row of the table identifies the bonds labeled R, S, and T? Let's go and see. We have R, we have S, and we have T. R is this one right over here. S is this one here, and T is this one here. So R cannot be covalent. R cannot be hydrogen. Therefore, it is a phosphodiester bond right over here because this is a phosphate group. Therefore, it's going to be a phosphodiester bond. And S is a covalent bond, as you can see, because it's between 2 carbon atoms. And T is attached to organic group. We have one organic group here. We have another one over here. Therefore, it is going to be a hydrogen bond. So yeah, the answer is D. Name 2 types of gene mutation. That's pretty simple. It's a pretty easy answer. We have substitution, we have insertion, and deletion. These are important. Remember them. It's pretty easy marks, and it's easy to remember. So yeah, let's move on. The graph shows the estimated mutation rate in 4 groups of primates. Okay, we have over here mutation rate per base pair, and we have the mean age when breeding starts. We have a key right over here to explain what's going on and which data represents what animal. They're asking us to describe 2 conclusions that can be made about the mutation rate in these 4 groups. So the first pretty easy conclusion to see and understand is that there was the highest mutation rate was found in prosimians. Prosimians right over here. And these are the prosimians. and they are the highest, the highest ones compared to the other groups. The second conclusion that we can make is that apes are the only primates that aren't affected by mutation within the beginning stages or ages of breeding. We can see that they start getting affected much later on than compared to the other groups. Now I have question 3. Question 3 is about protein synthesis. It's about DNA, RNA, start and stop codons, ribosomes, which is another extremely important subject. And they ask questions about these. They ask these questions not just in Unit 1, but also in Unit 2, sometimes 4. So it's really important that you remember your protein synthesis, DNA, and the enzymes. So yeah, let's start with this question. It says, describe the role of RNA polymerase in protein synthesis. So RNA polymerase is an enzyme, and what it basically does is it converts DNA, it doesn't convert, but it changes DNA into RNA, which is known as transcription. So I wrote, RNA polymerase transcribes DNA to produce RNA. RNA polymerase binds to the DNA molecule and unwinds the DNA helix, separating the DNA strands. RNA polymerase also lines up the RNA nucleotides on the template strand and forms a phosphodiester bond between the RNA nucleotides. RNA polymerase also repairs mistakes in the formed RNA. So yeah, it's pretty simple. Honestly, it's really easy to answer these questions if you have a model answer, which simply describes the whole process. That way you can memorize it and you can remember it, and if the question comes, you can just write it down. So yeah, just remember to write this down somewhere and to remember how to answer this question. Over here, I included not just one point, I included a few points. I think I got the whole concept, hopefully. Now we have compare and contrast the structure of the start and stop codons and how they work. So similarities, both codons consist of 3 bases. However, let's move on to differences. So start codons start translation, whereas stop codons, they end translation. That's pretty easy to remember. Start codons have 1 possible codon, but stop codons have 3 possible codons. tRNA binds to only start codons. So the tRNA molecule, they bind only to start codons. That's another difference. And finally, we have start codons code for an amino acid, whereas stop codons do not code for an amino acid. Those were the differences. Let's move on. Describe the roles of the ribosomes in protein synthesis. So what do ribosomes do in protein synthesis? Ribosomes synthesize polypeptide chains by translating genetic information from mRNA into polypeptide chains, which are the bases of proteins. Ribosomes hold 2 tRNAs together until a peptide. Okay, now moving on to question 4. Many animals have a heart and circulation. The diagram shows a human heart and its major blood vessels labeled P, Q, R, and S. Okay, here we have the diagram on the first question. So which pair of blood vessels transports blood to and from the lungs? As you can see over here, I labeled the diagram. So over here we have Q, which is the pulmonary artery. And here we have S, which is the pulmonary vein. Since it is pulmonary, it's related with the lungs. Therefore, the answer for the first part will be Q and S. Now, we're being asked, which blood vessels have valves along their length? So just to remind you, this right here is a valve. And the heart has 4 valves, right? So this first one, second one, third one, fourth one. 2 valves in each side of the heart. Therefore, now we have to see which labeled part has a valve, and that is going to be P, right here. We have this one right over here. However, it's not labeled. We have this one here as well, not labeled. We have this one here. It's not labeled. So the only one that does have a valve that is labeled is P. Therefore, over here, as you can see, the answer here is P. Which blood vessel does the coronary artery branch from? We know the coronary artery branches from the aorta. Sorry, let me write that better. Yep, the aorta. That's not any better, is it? But yeah, right over here, the aorta, and the aorta is labeled R. So the answer is R. Now, capillaries enable the movement of molecules between the blood and cells, okay? So the distance the molecules diffuse between the blood and cells affects the rate of diffusion. This is known as the diffusion distance. They asked us to name 2 other factors that affect the rate of diffusion. Now we have surface area, we have temperature, we have concentration gradient. So the larger the surface area, the greater the rate of diffusion, right? So greater temperature, greater rate of diffusion. Greater concentration gradient, greater rate of diffusion, right? We know that when it comes to temperature, it has to do with the kinetic energy. A raise in temperature means that there's a raise in kinetic energy. Therefore, the particles will be moving much faster. Therefore, diffusion will occur at a quicker rate. Surface area, if there's, a larger surface area, then that allows the molecules to move from a higher concentration to a lower concentration much more quickly than compared to a smaller surface area. And the greater the concentration gradient, the faster the rate of diffusion. Now they asked us name the law of diffusion that can be used to calculate the rate of diffusion, and that is right over here, Fick's law. Now we have this question over here. In the beginning, it may look like it's complicated. What I did was I saw like a lot of numbers and I got panicked and I just started I started with ratios. I don't know why, but I didn't read the question properly. That's why it's important to read the questions properly. Once I took a second to read the question, I understood that, okay, this is actually a very easy, simple question. We just have to convert the values that are given to us to find the answers, right? So let me explain. Now here we have 1 micrometer. Here we have 10 micrometers. Here we have 100 micrometers. Here we have 1 millimeter. What we need to do is we need to convert the millimeter into micrometer, which we can do that by multiplying it by 1,000. That's pretty easy. Right over here, I've written it down. 1 millimeter is equal to 1,000 micrometers. And then we have 0.5 milliseconds right over here, right? And then we have milliseconds again. Here we have seconds, so we have to convert this seconds into milliseconds, which we're going to do by multiplying by 100. Now, if I want to convert minutes into milliseconds, I've written it down here. What we have to do is we have to multiply by 60 and then multiply it by 1,000. And if we do that, we do 8.3 times 60 times 1,000. That gives us 490,000 milliseconds, which is approximately 500,000 milliseconds. So that right there is the answer to part C. Now moving along, we have explained why it is important for body cells to be close to capillaries to meet the demands of respiration. Use the information in the question and the table to support your answer. Now, we can see that a shorter diffusion distance results in a faster rate of diffusion. So, as we can see in the table over here, we can see here that 1 micrometer takes 0.5 milliseconds and 10 micrometers takes 50 milliseconds, which 50 milliseconds is a little too late to meet the demands of respiration, right? Because when we're respiring, not only are we taking in oxygen, we're also getting rid of the toxin, which is carbon dioxide, right? If we don't respire in time, or if we're not able to get rid of the carbon dioxide in our body within time, we start getting poisoned by carbon dioxide, because again, it is a toxin. Therefore, it's extremely important to remove or to get rid of this toxin in time. So, yeah. So a fast rate of diffusion is needed to ensure that cells are supplied with enough oxygen. So, a fast rate of diffusion is needed to ensure that cells are supplied with enough oxygen for respiration, and it allows carbon dioxide to diffuse out of the cells fast enough to avoid toxin buildup. So that's pretty much the answer for question 4. And the next part will be the other half of the exam, which is from questions 5 to 8. I hope this video helped you out. If you want me to solve a specific paper or a specific question, you can mention it down in the comments and I'll happily solve it for you. I'll happily explain things to you. Or if you want me to explain any chapter from the book, let me know in the comments and I'll help you out, because I'm here to help you out. I studied A-levels by myself at home. I didn't have any tutor. I studied everything by myself. So I'm here to help you, to share some important information and tips and tricks and just help you with your exams. I hope this video was beneficial for you. And yeah, all my answers are handwritten, because handwritten information retains into your memory much faster and it stays there for longer, which is why I'm taking the time out to actually write down the answers, and I'm taking the time out to write down model complete answers so that you don't need to open the marking scheme and to look at the marking scheme and then waste so much time just trying to write your own answer, because it's mentally taxing, it's tiring, it takes time. So I'm here to help you just really quickly get through the whole paper, to help you revise the chapters so you can remember the key important points. Again, yeah, thank you so much for watching. I appreciate it. If you want to tell me anything, you can let me know in the comments. Like if you have any advice to improve anything, like if you want me to change or do something, let me know. That's all for today. Bye.